All right, pre-AP biology. Today we are going to start um, a topic called population genetics. So this video is mainly for you guys that are at home, if you're quarantined or you're virtual or whatever, but also if you were in class and you saw my lecture, if you want to watch this as a review, you're more than welcome to do that. All right, so let's just kind of start off today with sort of a definition of what population genetics is. Population genetics is basically just applying our genetic principles that we've learned for the last month or so. Heterozygous, homozygous, punnett squares, all that stuff, and applying that to instead of an individual, we're applying it to a whole group of individuals, a whole population of organisms. So let's kind of review a couple of terms that we haven't really looked at in a while, just because I think it's a good time to review these terms, and also it'll kind of help you understand what a population is. So you guys might remember a species, right, is a group of organisms that can interbreed together. So for example, I don't know, zebras, right? Zebras can breed with each other and produce fertile offspring, right? That means the offspring one day can go ahead and reproduce on their own. So a zebra and a camel, they can't reproduce. They're not members of the same species, right? Now, a population is a group of organisms from the same species that lives in a specific geographical area. So, zebras, in the Serengeti of Africa, that would be a population that we can study. Now, we're going to look at a very specific species today. We are going to look at rabbits, little cottontail rabbits, so cute little fuzzy bunnies, right? Now, when we look at cottontail rabbits, when we look at any species, if we're going to study population genetics, we have to have a trait that we study. So in this case, we're just going to study fur color. We're just going to keep it nice and simple. Okay, brown dominates white. This is just a regular autosomal dominant trait. So not sex linkage, just regular dominance. Okay, no tricks or anything here. We'll keep it as simple as we can. Now, if we're going to study population genetics, we got to study a population. So what we're going to do is we're going to look at a population of rabbits in Crown Point because the area matters. We could also, I guess, study a population of rabbits in Maryville, right? But that would be a completely different area. That would be a different population. So the population is defined as the species and the area. So any of the rabbits within the town border of Crown Point, that's what we're going to end up studying. So studying population genetics is kind of interesting because you might be asking yourselves, well, how are we going to study all the rabbits in Crown Point? I mean, can we actually do that? Well, I mean, the answer to that is an obvious no. I mean, there's no way that we can go out. Let's say we did a class study and all the kids in our class went out into Crown Point, over the weekend, and I said to you, go out and capture every single rabbit. I mean, there's no way that you could do that. Right? It's impossible. So instead, what we would have to do is we would have to capture a sample of the population. Now, population sampling is basically just using a portion of the population and allowing that portion to represent the entire population. Now, the one thing about population sampling is it has to be random. So, for example, if I said, okay, this weekend, you know, we're going to go out into the town and we're going to capture as many bunnies as we can, as many rabbits as we can get. So you wouldn't want to capture them all, from like one area of town. You wouldn't want to get them all from, let's say, the northwestern part of the town. You would want to get these rabbits from all over the place. You want to get them, as randomly as you can, but you want to get them from as many different spots and areas in Crown Point as you can, and we're going to let that be the sample. So let's just assume, just for this example today, that we do this. We go out into town over the weekend, and you guys capture 100 rabbits, and you bring them back, and we study them. Okay, you don't hurt them. We keep them in little cages, and we take care of them. We feed them, but we capture 100. I mean, I think that's a doable thing. I mean, if we really had to do this, we probably could go out, and I guess we could set rabbit traps and maybe capture 100. But again, this would be a random sampling of rabbits that we would capture. So these 100 rabbits would represent the entire population of rabbits in Crown Point. Now, there might be 5,000 rabbits in Crown Point. Okay, so I mean, this little tiny sample, I mean, does it really represent the whole population? I mean, no, it doesn't, but this is the best way that we can do it. But you can probably see why a larger sample would be more accurate. I mean, if we went out and we captured 3,000 rabbits, our data would probably be a lot more accurate than, maybe only capturing 100. But if that's all that we can manage, then that's all that we can manage. So we're going to use these 100 rabbits again to represent the whole population. Now, there's a term here, it's called the gene pool. Maybe you've heard of this term before, but you maybe don't know what it means. It says here the gene pool is a term used to describe all of the genes or all the alleles, right? Those are the types of genes for a particular trait amongst all the members of a population or of a population sample. So if we collect 100 rabbits and we're studying their fur color, right? Think about it. How many alleles for fur color does every rabbit have? Every rabbit has two, right? Could be two dominants, could be two recessives, could be one of each. So if there's 100 rabbits, we can make the assumption that there are 200 alleles that we're going to be studying in this population sample. So let's say we get these 100 rabbits, and this is just how it breaks down. 30 of those rabbits that we capture are homozygous dominant, which means that they're brown, right? But they're big B, big B. 50 of those rabbits that we capture of the 100 are heterozygous, which means they're big B, little b, but they're also brown. 20 of the rabbits that we capture are homozygous recessive, which is little b, and those rabbits are obviously white. But again, we've captured 100 rabbits. That means we have captured 200 alleles. What we need to figure out is how many of those alleles are recessive and how many of those alleles are dominant. And if we know those things, we can figure out something called the allele frequencies. So let's go ahead and take a look again at these rabbits and these rabbits that we collected. Right? We said on the last slide that 30 of our rabbits were homozygous dominant, right? They were big B, big B. Well, that means that we've captured 60 dominant alleles. Those 30 homozygous dominant rabbits have 60 total dominant alleles. Now, remember, we said we captured 50 heterozygous rabbits. Those rabbits are big B, little b. So that means each rabbit has one dominant allele and one recessive allele. So those 50 heterozygous rabbits are contributing 50 dominant alleles and 50 recessive alleles to our gene pool. The gene pool is all the alleles for the population. Then we said that we had 20 homozygous recessive rabbits. So those rabbits contribute a total of 40 recessive alleles to the gene pool. So now this is a good point to just double check, right? 60 alleles plus 50 plus 50, right? This is the heterozygous rabbits, plus 40 equals 200. So these 200 alleles, this is kind of how they're broken down. Now, of those alleles, how many of them were dominant? Well, 110. These 60 here are dominant alleles, and these 50 here are dominant alleles. So 110 out of 200 are dominant. How many are recessive? 90, right? I mean, there's only recessive alleles left. So again, these 50 here are recessive, and these 40 here are recessive. That gives us a total of 90. Now, what we're going to do is we're going to use those values to determine something called allele frequencies. Allele frequencies basically tell us how common these alleles are in the population. And we can write allele frequencies either as decimals or percentages. You will need a calculator for this, so go ahead and take a second if you don't have one to go grab one. All right, so the first thing we're going to figure out is something called the frequency of the dominant allele. Now, we use a variable called P to represent the frequency of the dominant allele. I don't know why they do that, but they do. So again, in our rabbits, we have 110 out of 200 alleles are dominant. So all you simply do is just divide. All right, and that gives you this decimal, 0.55. Now that decimal as a percentage is 55%. So what that tells us is that all the genes in the gene pool, 55% of them are dominant. If you threw all of these genes into a hat and reached in and grabbed one, there's a 55% chance that gene is going to be a dominant gene. That's what that tells us. Okay. So the frequency of the dominant allele in this case is 55%. But again, you can write it as a decimal or you can write it in percentage form. All right, I would prefer that you wrote it as a decimal, and you'll see why that is in a minute. Now, we can also figure out the frequency of the recessive allele, right? 90 of our alleles were recessive. So simply just divide 90 by 200, and you get 0.45 or 45%. So we assign this variable, it's called Q. All right, Q is the frequency of the recessive allele. So in this case, Q is 0.45 or 45%. So again, if we took all of our genes, put them into a hat, and I reached in and I picked one, there's a 45% chance that that allele is going to be a recessive allele. Okay. So I'm going to stop this video here today. You'll go ahead and look over that, watch it again if you need to. But I think this is a good point to stop and take a break, and then we'll watch the second video.